We have 5 main tastes: bitter, salty, sweet, sour, and glutamate. Each one depends on a different G protein receptor. Bitter, sweet, and glutamate are the G protein receptor. Salty and sour are the ion channels that go through the G protein receptor. This is the tongue. There are 3 different types of taste buds that are on your tongue. The first one is fungiform. Fungiform is the taste bud that's actually on the tip of your tongue, and when you go to taste food, that's the first thing that it will taste. The next one is foliate. Foliate is these little spikes right here that are on the edge of your tongue that sends a signal to your taste buds about what you are tasting. Circumvalent is the actual bud, which are these little blue things right here. Each 5 different tastes are included on the bud. They can be in any order, but they have to be side by side so they can send a signal to each other. So this is a taste bud, and the taste bud is connected to any one of the 5 tastes over here. And when you have a taste bud, let's say this little green part right here is sweet. The sweet part will go through these little connectors right here. The sweet will go through the taste buds right here, and it will send a signal to the brain. Let's say this is the brain right here. It will send a signal to each part and say this part is sweet, this part is bitter. It will send the sweetness from the taste bud. So this is the sweet, and when the sweet part right here, like the green part right here, sends a receptor signal through the taste bud and into the brain, this brain right here will take the signal and place this sweetness of the food into the section of where the sweet is. So taste is another word for gustation. And gustation is the whole thing right here. This taste bud is the gustation. And when it interacts with the tongue and the food, it sends a signal to the brain letting you know what kind of food you're tasting. This is known as the axon, which is a spiky thing on the edge of your tongue. So let's say this is the nucleus, and this whole thing right here is the sweet taste bud. Let's say glucose is on the edge of the sweet cell. When you eat something, it will activate right here. It will activate the little hairs on the edge of your tongue. The glucose will then interact, and it will go through the cell, interact with the nucleus, and go to the end of the tongue, which will send a signal. So this is within the cell membrane. So let's imagine this is the cell membrane. This is the receptor where glucose is made. Let's imagine that glucose binds to ligand. And when glucose and the receptor bind to ligand, it crosses the little section right here, which is called the boundary, and it makes G protein. And when G protein is produced, it opens the ion channels, which is salty and sour. And when it opens ion channels, it makes AP. And when AP goes to these little cells right here, it makes NiCi. And when NiCi is interacted or bonded with these little cells right here, it makes a positive reaction. The positive reaction goes through the cells without interacting, and it produces AP. When that AP is produced, it sends a signal to the brain telling you what you have ate and how it's digested and what is it tasting like. It could be any one of these: bitter, salty, sweet, sour, or glutamate. It's most likely to be bitter in the cell, this part right here. And this part has to do with this part over here, but that is within the cell. So let's say this is the sweet cell right here. This is also called the axon. This is the nucleus that contains sugar, and the sugar activates the cell. The NiCi tricks the brain into thinking that it's sweet, and the NiCi, when it tricks the brain into thinking it is sweet, it can go into the cell, and it mimics the sugar and it is allowed into the cell. And when it's allowed into the cell, the sugar and the sweet mix together and they bind together, and they make gustation. And gustation is the main taste that is sent to the brain.